Hello, everybody, and welcome to Chapter 2. In the last chapter, we went over the brief history of a bit of psychology and of neuroscience slash biopsychology. We talked about the nature-nurture question. We talked about monism versus dualism. And we left off sort of on a cliffhanger because we talked about, or I talked about at least, how the nervous system communicates with electrical activity. But what I didn't go into is the fact that that's only half the story, and that what researchers originally discovered is that these neurons are not connected to each other like wires in a circuit, but rather there's a little tiny gap in between them where they don't touch, and that is sort of where the magic happens. So the electrical activity is true. It's been researched and proven for hundreds of years, but it's that other portion of it that I really wanted to get into. So we're going to talk about both the electrical activity, what causes it, how we generate our own electricity, as well as what happens after we that electrical signal gets to the end of the neuron. Well, basically what it does is opens the gates and allows the chemical activity to go through. So we're going to go over all of that in this one. Chapter 2 is a little intense. It's a little specific, so strap in. Normally I use in class 3, I take 3 regular class times to go over this chapter because I go over some of the stuff again and again. But since this is a recorded version, obviously I'm not going to do that. But it is up to you if you want to watch it more than one time. You can also read from the written out lectures. So here I present to you Chapter 2, Communication within the Nervous System. I am going to break this up into 2 lectures because nobody needs all of this in one long one. So we'll talk about the cells that make us who we are and then how neurons communicate with one another. And I'm hoping that this time around, I will get cooperation from my computer. So our nervous system, remember one thing I said, I hope you remember everything I said, but one thing I said last time is that everything that we do, every thought, every urge, every spontaneous thing, every reflex, every thing we say, everything we think is within our brains. And that means that the brain is in charge of everything we do. It's 20% of our entire energy output for our body. In that sense, we have over 100 billion neurons in our nervous system that make up our nervous system, and that's our communication system. But you may have heard this term, which I absolutely despise, that we only use about 10% of our brain. That drives me insane. I'm sorry there's no limitless pill that you can take that'll make you move things with your mind or anything like that. But what you'll see after this chapter, and we're going to skip to chapter 5 after this one, you'll see why, and I'll explain it. Then we'll go back to chapter 3 where we talk about the specific localization, the specific functions of the brain, and you'll see that we use the entire brain. The only thing that I can think of is that maybe people think we're only using 10% of our memory storage, or the fact that our nervous system is only made up of 10% actual neurons and brain. The rest of our brain volume is actually made up of helper cells that their sole job is to make sure that the neurons survive so that we can communicate and that they're as healthy as possible. These are called glial cells, or glia, and that makes up about 90% of your brain volume. So maybe it's that people say we only use 10% of our brains. I don't buy that. 10% of our brain volume? Sure. Anyways, let's talk about this essential building block of the nervous system, of which we have over 100 billion. These are called neurons. Sometimes they're called brain cells or nerve cells, but we refer to them as neurons. And neurons have a few specific functions. One, remember that we don't get information about our outside world from the five senses until these cells, these neurons, bring this information up to the brain from our body. So what we see comes from our eyes, that goes to the brain to get interpreted. What we hear from our ears, what we feel or what we're touching from our sensors in our skin, including pain and temperature. That's in all of our skin cells and even some of our internal organs, so that we know if something is hurting. And taste and smell from our nose and tongue. All of this information goes from these senses through these neurons to our brain to get interpreted. And then once our brain interprets it, it makes a message, figures out what the message is, the big picture. Then it sends commands back down to the body. So conveying sensory information to the brain, going up to the brain, sending commands back out to the body is that last one. So that's going down to the muscles and glands and all of that stuff. And there's also neurons that connect to each other that carry out the operations involved in the thought process, feelings, emotions, memories, which ends up being a whole circuit, which we'll go over in chapter 12. So there are there are many different things that the neurons are responsible for. These are the things the main things. There are three main parts of an axon. Of a neuron. It is probably here where I tell you that most of the things that are named in the brain and in for body. And there is the axon. Dendrite is Latin for tree branch. And so out of the cell body, which is the main life force of the cell that contains remember we talked about every cell contains your DNA, a copy of your genetic code. So in that cell body is what's keeping your cells, your neurons alive, and also has a copy of your DNA. The dendrites stick out straight out of the cell body, and they look like little tree branches. The dendrites are actually the ones that are picking up that chemical signal. I told you we have the electrical and the chemical signal that these neurons are not touching. So in the space between the neurons, the dendrites are picking up the chemicals. They bind to the dendrites kind of like a lock and a key. So dopamine will only bind to a dopamine receptor on the dendrites, glutamate to a glutamate receptor, serotonin to a serotonin receptor. So they are responsible for basically picking up the signal. But what about this very important electrical activity? Well, that's that last part where you see the axon down here. The axon is the long part of a neuron that carries the electrical signal. And that electrical signal is very important because it has to remain strong all the way through. Some axons can be tiny, and some axons can go all the way from your spinal cord down to your little toe. Now imagine if you're Shaquille O'Neal, who's like 7'2". You need to be able to move those feet, right? Especially during his basketball days with the Lakers. So that axon is extremely long in an over 7-foot tall person. And we need to make sure that the signal goes from the top of that neuron all the way down the axon to be able to release the chemical. So sometimes the neuron needs help, and that's where those glial cells come in. These axons are a very long portion of the neuron that carries the electrical signal, but they need some insulation. So these axons are insulated with this fatty tissue that's whitish in color called myelin. Myelin is made by one of the glial cells, or one type of glial cell. And at the end of the axon are these little terminals, or these little things that look like buttons, that are called axon terminals or terminal buttons. And those have the neurotransmitter or chemicals in our brain that release them into that space in between the neurons. So there is a little bit of space between these axon terminals and the dendrites of another cell, and that chemical has to be released from the terminal or terminal button and get picked up by the next cell, which is the dendrites. So let me, it's probably better if I give you a visual, if my computer will let me. Maybe I need a hard reset on this thing, but either way, I can make this easier. Once again, back to presentation. So here, once it comes, is basically a neuron. Now this is a specific type of neuron called a multipolar neuron. We do have unipolar and bipolar neurons, as I'll get into. But the main type of neuron, especially the one that connects, as you can see here, and you can tell what type of neuron this is because of what it connects to. So multipolar means that there's many spots. These are the dendrites. Some of them are just right here on attached to the cell body itself. Here's the cell body with the nucleus. That's your life of the cell. That's your DNA, all of that stuff. Here are the dendrites, and again, under a microscope, you can see why they're called dendrites, because they look like little tree branches. Down the long portion of this neuron is the axon, and the axon is connecting here to a muscle fiber. So this must be what we call a motor neuron. Motor neuron means this is coming from the brain and spinal cord to a muscle to tell it what to do. Let's say that I'm playing dodgeball and a ball is coming rapidly at my face. My eyes will get the information about the ball coming closer to me, send it to my brain. My brain will then have to make a decision of what to do. It will send information through the motor neurons to the muscles to tell me to either duck, to move out of the way, to put my hands up so I don't get bombed in the face, or if it's too late, then I'll have another group of cells that tells me that hurt, which will go up to the brain, right? So here we see this portion of the neuron called the axon, and you can see that it's sort of covered in a fatty tissue. This is the myelin or myelin sheath, like a sword. You put the sword into the sheath, right? And although you might think that it covers the entire span of the axon, I like to use the example of like a phone charger or any sort of computer cord that's covered completely in that plastic, which does two things, because the electrical activity, again, from the wall to the computer or from the wall for your charger to your phone, is that it ensures two things. One, that you're not going to get electrocuted because you don't want to touch bare wires. That's not a good idea. Two, is that it ensures that the neuron signal or the electrical signal gets to where it needs to go. Now remember, this is a much shorter axon than we would see in, again, Shaq or somebody really tall in their nervous system going down to their little toes, which are actually big toes in comparison to me. But the idea behind it is that the signal, which starts basically here at the axon hillock, where the cell body meets the axon, because that's the lowest point of voltage to get an electrical charge, it goes down the axon, and again, here's the terminals. We have to ensure that the signal is just as strong here at the end to release the chemicals as it was at the very beginning when it started. So that's why, right? That's the advantage of a cord that's covered in that, plastic coating or whatever, is that not only does it not electrocute you if you touch it, but it makes sure that the signal goes in one direction in the way it's supposed to go. We don't want electrical signal going out the sides. We don't want it propagating backwards towards the cell body, because that doesn't do any good for these terminals, which are going to release the chemicals onto the muscle or to another brain structure or to another cell. So we have this myelin covering that's made by glial cells that ensures that the signal A goes a lot faster because it needs to be efficient, and B, that it doesn't go backwards or out the sides, because again, we need it to be just as strong at the end as it is at the beginning. But you may notice that instead of being connected fully, like the cord for your charger or your computer, there are little holes or little bare spots of axon on these underneath the myelin. And that actually is advantageous. They're actually more like hot dog links almost, right, than a fully covered cord. We're going to get to that in just a few minutes. But the nice thing about it is, like your phone, when the battery gets low, it recharges itself. These little bare spots of axon here actually allow that electrical signal to do just that. The signal goes and gets faster through the myelin, and then when it hits the bare spot, it basically recharges itself, and you get a new, what we call action potential. And it recharges, and it recharges, and it recharges. Therefore, when it gets to the end here, it is just as strong, it's been recharged, as it was when it started. And if the signal's strong enough, as we'll talk about, that's why you can get the chemicals to release efficiently. Again, those glial cells that make up 90% of the brain are making sure that these neurons are healthy, that they have energy or food, that any debris or bad stuff gets nursed back to health or eaten away, and to ensure that the signal is very strong. I apologize here for the lack of picture. Sometimes, I don't know why on my computer that some of the slides are missing the picture. You can see that here, but I will tell you the saving grace of that is when you review the material written out, every single slide and even more pictures than the slides are available there. So this just shows that there are 3 different types of neurons. It shows what the other ones look like. That one on the previous slide was called a multipolar neuron. There are also 2 neurons called a unipolar and a bipolar neuron. Now, I'm not going to, on the exam, show you a picture and tell you to label which one is which. Just know that we use the multipolar as our example throughout, basically. This picture, and when you do go through the written portion of this, you will see the unipolar and bipolar picture from here. But the multipolar neuron is the one that we use the most, so that's going to be our representative neuron. Just know that there are 3 types: unipolar, bipolar, and multipolar. And of course, this one worked so well, and now it won't. So let's go to the next slide. So just like there are 3 different types of neurons, unipolar or bipolar are actually the only ones that are what we call sensory neurons. So remember we talked about neurons that bring sensory information from the 5 senses, from our outside world, to the brain to be interpreted. In chapter 3, of course, we'll talk about where these neuron circuits go, which part of the brain specifically to interpret which sense. And then we'll really get into the visual system and auditory system in the 3rd module. In the regular semester, we also have the touch system. So, motor neurons are multipolar, as we saw in that slide a couple slides ago, and that's output from the brain and spinal cord to the muscles or organs to make our body move in what direction we need it to, or duck if a dodgeball is coming at our face. The unipolar or bipolar neurons are sensory neurons. Those are the ones that, depending on which sensory system, will tell us what's happening in our outside world. Those go up to the brain to let the brain know what we are seeing, hearing, smelling, tasting, and touching, or what is touching us. Touch also includes pain and temperature, so if something hurts or if something is too hot or cold, it lets us know. Those are the unipolar and bipolar neurons. And then the one we actually have the most of in our system, because again, we don't just have neurons that go straight to the muscles or straight to another structure, but we have a lot of neurons that communicate with each other, especially when we're trying to combine motor and sensory information. If sensory information has to go all the way to the brain, and if motor information has to go all the way down to the muscles, sometimes they need to coordinate with each other so that things can happen as quick as possible. So interneurons connect neurons to neurons, and they integrate motor and sensory information so that we're getting stuff at the same time. This is a lot of times what happens with reflexes, or if you, and I think I'll explain this a little later, but if you have ever touched a hot stove or a hot pot that you didn't realize was hot, you may have noticed that your hand in a reflexive action moves away from the pot as soon as you realize it's hot, but it's not for another half a second or second later that you actually feel the pain. Well, there's an interneuron that intercepts the information at the level of the spinal cord between motor and sensory, and it allows your reflex action to start when the information hits the spinal cord. But pain is interpreted all the way up in the brain. So it isn't until that information gets all the way up to the brain that you actually feel the pain. That's why you can reflexively move your hand away and you won't feel the pain of that hot pot or hot stove for about a half a second to a second later. There you go. And you just turned without my, oops, geez. This is such a task sometimes. Looks like we're going back manually once again. I really need to get this looked at. Okay. Pardon me for just a second. So now that we know what the neurons do, it would be really nice to understand how the neurons work with electrical activity by looking at the structure of the cell so we can understand how electrical activity is even possible. So here is a microscopic view of basically the layer, the membrane of a cell. And what we see here is that the cell is actually maintained by two layers of lipid or fatty molecules that are opposite one another. And so it's a double or bilayer for the membrane. What this allows is for certain things like water and gases to freely pass in and out of the cell. So this is the inside of the cell down here. This is the outside of the cell. The only other things that can get in are molecules through these little channels. This kind of looks like a half of a green pepper. Dang it. Sorry about that. I really need to just keep my hand off the mouse, apparently. Or mouse pad. So let's go back. My apologies. This is going to be a frustrating event if I can't get under control. All right. So here we see this lipid bilayer. So there's two layers of fatty molecules that maintain the composure. They maintain the shape of the cell. So again, water can pass through, certain gases can pass through, but any other molecule can't get in or out unless it goes through one of these little channels that looks like sort of the green pepper thing. And like a lock and a key, that molecule has to be able to fit into this channel in order to get in, and can only get in if this channel is actually open. It has a gate on it, kind of like the gate to your yard or to your apartment complex or something. If it's closed, nothing is going to come through except for, again, maybe a little bit of water or gases. But the actual molecules have to be able to go through these protein channels. So what that means is these proteins allow the cell to be what we call selectively permeable, which means they can let things in or out, but only on their terms. As you see that the lipid molecules have little heads on them, and the heads are facing opposite of each other. So the heads are facing the inside and the outside of the cell, and the tails that don't like water, because there's fluid or water inside the cell and outside the cell. So the heads are what we call hydrophilic. They really like the water. The tails are facing each other in the middle of this bilayer. They don't like water. They're repelled by water, kind of like magnetic forces where two negatives repel each other, right? Or two positives repel each other. So these tails don't like water. We call them hydrophobic, like they're afraid of the water. So you have the heads of the molecule on the inside of the cell and the heads facing the outside of the cell. And the tails are facing each other in the middle. So it's able to hold the cell shape and hold the cell together, as well as controlling what goes in and out of the cell. And watch, now it's not going to change because, oh, good. I was going to say because it's being moody. So what this says is then the neuron is selectively permeable. Permeable means that things can pass through or in or out of it. Selectively permeable means that things can only pass if they fit through those channels and at the times the channels are open. So again, water and gases can pass freely, but any other substance, any other molecules that don't fit in those little channels cannot get in. They cannot compromise the integrity of the cell. They will not come in unless they can, unless those channels are open and the cell allows them to come in. So the others pass through those protein channels, but only when those channels are open under certain circumstances. What are the circumstances in which those channels are open? A lot of times it has to do with voltage or electrical activity. So when there's a certain electrical charge or electrical activity, then those cells can, or those channels can open. The cell is open to some of these molecules, and the molecules can either come in or come out of the cell. What this does, because these molecules bring a positive or negative charge with them, is it changes the voltage level. It changes the electrical activity of the cells. So this change in voltage from the cell being open or closed and allowing certain molecules in, this is what we call polarization. So it results from this permeability of the membrane. So the definition of polarization is the difference in charge. So we need to know a specific measurement of voltage. It's the difference in electrical charge between the inside and the outside of the cell. So in this case, we take an electrode. If you're wondering how we measure this, you take one electrode and put it inside the cell. You take one electrode and put it outside the cell in the water or the liquid. And then you take the difference. Whatever the difference is inside the cell minus, right, outside the cell is going to be your voltage. So this difference in electrical charge is referred to as the cell's voltage level. And any potential, we use the word potential, local potential, action potential, you're going to hear this word a lot, represents a change in the voltage. If it's a small change that doesn't cause a whole chemical reaction or electrical reaction, we call it a local or graded potential. If it is enough to cause the cell to actually fire an electrical signal all the way down that axon, activate the cell, put it into action, we call it, that's right, an action potential. I will get into the difference there and what that means. If this will let me change. Nope, I guess we're back to manual. Again, my apologies for the jerkiness of this. I'm going to just say my computer's being a jerk. So, when nothing else is happening, when all we have is the resting potential or the difference in electrical charge when there's nothing moving across. If the cell is sort of turned off temporarily and those channels are closed, when we measure in between the inside and outside of the cell, this is called the resting potential. It's basically defined as the difference in charge between inside and outside of the membrane when the neuron is at rest or when nothing is happening, when there's no movement of molecules or ions in or out of the cell. Now, again, I told you we have 100 billion neurons, and I told you we have 3 different types of cells: unipolar, bipolar, multipolar, sensory, motor neuron, interneuron. So there are different voltage levels, resting potentials for the different cells. I also told you that our multipolar cell is going to be our main example. So even though these cells are generally between -40 and -80 millivolts. That's a millivolt is 1/1000th of a volt. Yes, when you think of the electrical charge in a wall, that wall plug is about 110 volts, regular volts. This is millivolts, right? We don't want too much electrical activity in our brain, otherwise we'll electrocute ourselves. And if you've ever seen anybody having a seizure, it kind of looks like they're being electrocuted. Well, that seizure results from too much activity. We'll go over that. So between -40 and -80 millivolts. That's right, it's negative inside the cell with respect to outside the cell. But for the purposes of this class, for the purposes of having one specific number that we can go to, the resting potential, and you'll need to know this for, quiz or exams or things like that, the typical resting potential number that we're going to use for a typical multipolar neuron is around -70 millivolts. So you stick an electrode inside, you stick an electrode outside of the cell, and the difference in charge outside is a little bit more positive. Inside is a little bit more negative. Why would it be negative inside the cell and not outside the cell? Or less negative, right? Outside. Well, that's because of the ions or the molecules that are both in and out of the cell. So there are molecules or ions that are already in the cell from the beginning of the cell, and there are molecules or ions outside the cell. And there are, right, those channels that open that allow things to either come in or out. When ions come in the cell, they bring a positive or negative charge, and that changes the potential of the cell. So there is an unequal distribution of ions on either side of the membrane. Sitting on the outside of the cell are a lot of sodium ions, like salt, Na+. Those are positive ions. There are also chloride ions, which are negative, and you can see by their symbols, Na+, Cl-, that they're either positive or negatively charged. Inside the cell, we have positive charged potassium, K+, ions, and here is the main reason why it is more negative inside the cell than outside. The biggest of all of these ions, the biggest of all these molecules are called anions. I know it almost looks like onions. It's anions, A negative. Those anions are too big to fit through any of those protein channels. And they are so big that they bring a lot of negative charge with them. So because of those anions stuck inside the cells, it is more negative inside the cell than outside. And there are a lot more sodium ions outside the cell than chloride. The anions are bigger than the potassium. So you get more negative inside, more positive outside, and the difference at rest is around -70 millivolts. So we're going to use that as our main number for resting potential. Come on, please. No? No. Okay. If you're going to be mean, you can be mean. Let's move down a little bit. Again, I apologize. Technical difficulties. So, what resting potential means here is, and again, this graphic will be on the actual slides in the actual typed out portion. But what it shows is sort of the membrane layer, and it shows what's happening with activity. So even at rest, there are a couple of channels that kind of remain open. So a little bit of sodium, which is outside, wants to come in. A little bit of potassium that's inside wants to kind of leaks out, and it may change the voltage slightly. But how do we maintain resting potential is that there's something called the sodium-potassium pump, which basically, if a couple of sodium find their way in and a couple of potassium try to escape and find their way outside, this little pump will basically put it back to normal. It'll move those sodium molecules back out, and pump those potassium molecules back in. So again, just to review, sodium is outside of the cell and potassium is inside of the cell. But as you've seen here, they kind of don't want to be there. They want to go to the opposite side of the spectrum. Why do you think that is? Well, there are actually two main forces that tell us why a molecule may want to change from inside to outside of the cell or from outside to inside of the cell. One of them you may have heard of before in biology class, called the force of diffusion, which is pretty easy to explain. And the other one is called electrostatic pressure. So sodium's sitting on the outside of the cell, but sodium doesn't want to be on the outside of the cell. Sodium wants to be inside, kind of like standing in line for an exclusive club, trying to get into this club or this party because you really want to be inside. So that's sodium. Sodium's outside and it really wants to be inside. Potassium, on the other hand, is in this crowded party. You know, sometimes you get into the bar or club and it's just wall-to-wall people. I'm obviously not now, but you know, you go to this party and it's just no elbow room. You're kind of, you know, stuck where you are, and it's really claustrophobic almost. So in that case, right, here, potassium, which is kind of stuck together next to these big anions and other molecules, wants to be outside. This is basically the form of diffusion. Diffusion says that ions have this tendency to want to flow from areas of high concentration to areas of low concentration. If sodium is stuck with a whole bunch of other sodium molecules outside, it wants to be in. Potassium, stuck with a bunch of other potassium molecules inside, it wants to go out and get some fresh air. So that's the force of diffusion. So automatically, sodium wants to be inside the cell because of less sodium around it. Potassium wants to be outside of the cell because there's less potassium outside. So that's one of the main forces. So both of those want to happen because of diffusion. At rest, a little bit of sodium trickles in and sneaks in. Maybe there's a hole in the fence, right? A little bit of potassium sneaks out, and the sodium-potassium pump puts them back to where they're supposed to go. So at rest, which is not very long, but if we were to freeze time, at rest, we are at negative 70 millivolts, and even with that little bit of charge leaving or coming in, it gets pumped back out by this sodium-potassium pump. We'll get back to that later. There is another force at work besides diffusion, but at rest, it's only going to be working for one of these molecules and not the other. We already know that both of them want to be in the other side because of diffusion, but the other thing that's really affecting sodium is what we call electrostatic pressure. In electrostatic pressure, what we see is that ions are attracted to the opposite of what they are. So positive wants to be in a negative environment, and negative wants to be in a positive environment. So with sodium on the outside of the cell, there's much more sodium outside, so it wants to be inside the cell because of diffusion, and sodium is a positive ion, and it wants to be in a more negative environment. So ions are attracted to the opposite charge. Positive wants to be inside the cell because the inside of the cell is negative. Remember, negative 70 millivolts. So sodium is really motivated. Sodium wants to be inside this party, inside this club, because of diffusion and because of electrostatic pressure. Too many of sodium's friends, too many other sodium molecules are outside, so it wants to be in, and it's positive outside and negative inside. So the positive ion wants to be inside. Potassium, on the other hand, is inside the cell and yes, wants to be outside because of diffusion. But there is no electrostatic pressure for potassium. Why is that? Well, if you haven't figured it out, I'll tell you. Potassium is positive. Potassium is already in a negative environment, and therefore, it kind of wants to get out because of diffusion, but there's no electrostatic pressure for potassium. So just to review, sodium has both diffusion and electrostatic pressure wanting it to get in, so it really wants to get in the cell, like badly. Like it's got to pee and the only bathrooms are inside. Potassium is like, there's a lot of other potassium here. I don't have any room to stretch out. I'd like to be outside, but I'm also comfortable because I'm positive in a negative environment. So there is diffusion for potassium, but there's no electrostatic pressure. Hopefully you guys are still following along. Okay. So now we have voltage wanting to change. We have potassium kind of wanting to get out because of diffusion, and we have sodium really wanting to get in because of both diffusion and electrostatic pressure, which means the first chance that any of these molecules gets to go in is sodium, because sodium has both electrostatic pressure and diffusion wanting to get in the cell. When the cell, when we get a signal and it causes a change in the cell, a lot of times these neurons fire a excitatory signal, which means voltage increases. And this causes what we call a hypopolarization. I like using depolarization. So I should have changed those because I'd rather say depolarization than hypo, because we have hyper, and so it gets a little confusing. So I like using depolarization better than hypopolarization, but it means the same thing. So when there's a excitatory signal, when there is an increase in voltage, we get a depolarization of the membrane. That means that the cell becomes temporarily more positive. It goes towards zero from -70, trying to get into the positive range. So again, this hypo or depolarization is caused by the change in ions, aka sodium comes into the cell. Sodium makes the cell less negative or more positive because it's bringing positive charge with it. If it changes only slightly, basically that pump is going to bring it back to resting potential. Our brain, our neurons like to be at rest. They don't want to have to work all the time. So if we get a little bit of sodium coming in, we get a little bit of what we call a hypo or depolarization, and it causes a little change in voltage, and this is called a local potential, but it doesn't go very far. So it goes in and then gets pumped back out. Nothing happens, cell goes back to rest. At the axon hillock, again, that's the point where the axon meets the cell body. That's where the voltage level can be manipulated the easiest. If this hypopolarization, if this depolarization goes up about 10 millivolts to -60, then it reaches what we call the threshold, which is kind of an imaginary line, and that activates the potential of the cell. So if you depolarize it by 10 millivolts and it reaches -60, then it causes the cell to actually fire an electrical charge all the way down that axon to release chemicals. This firing of this impulse or electrical charge down the cell, down the axon, is called, as I said before, an action potential. So if you only get a little bit of change, a little bit of depolarization, you're going to get a local potential. It's going to get pumped out, and you're going to go back to rest at -70. If, however, you get enough change, enough sodium coming in to change the voltage all the way down to -60 millivolts, then you get an action potential, and it sparks a whole bunch of different things in the process, which we are going to explain, or at least I am. We're going to do this. Oh, there we go. So, whoops, we're in the right spot. I jumped the gun. Here we see a voltage sort of meter, right? So here we see resting potential is here about -70. Here we see anything below that is what we call hyperpolarized, so making the cell more negative. In green, it's kind of hard to see on my computer. hypopolarized or depolarized, which brings it closer to, makes it less negative or brings it closer to zero. At some point, here we see if we only get a little bit of sodium coming in, we get a little local potential and then it goes back down to rest. If we get a couple of molecules that come in, maybe double that, we get a little bit more movement and then it's pumped out again. Here is this threshold at -60, right? At -60, if this threshold is met, the action potential is what we call an all or none process. If it reaches this threshold, if it reaches this voltage level, it will always, 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 100% of the time, fire this electrical impulse, which causes the voltage to spike way up above zero. That's what fires that electrical impulse all the way down the cell. If it's below the threshold, even if you have 3 inputs, right? As long as it's below the threshold, it will never fire. It'll go back down to zero. It'll remain a local potential, and it will never cause an action potential. And it doesn't matter if you barely hit the threshold or if your signal goes way above the threshold. As long as it hits the threshold or goes above, whether it barely hits or whether it's way up here, it's going to cause that action potential to fire in the same way every single time. The action potential, once again, is the way that that signal gets from the axon hillock all the way down to the axon terminals to be able to release the chemicals. So we call this an all or none phenomenon. Even if it's just below the threshold, if it doesn't hit the threshold, it will fire never, 0% of the time. If it barely touches the threshold or goes above it, it will fire 100% of the time. So we call this an all or none phenomenon. I'm going to try and spend the rest of the time today talking about what this electrical signal is, why it goes up, and why it comes down. And then you see here at the end, towards the end of this action potential, it actually goes below resting potential down to almost -75 or -80 before it comes back. Why does that happen? Why does it spike so big over here? What happens at the peak that makes it go back down? That's what we're going to talk about. And of course, looks like I'm going to have to do this manually again. We should all be used to this by now, right? So, once again, it looks like the, I apologize that the, some of these graphics are missing, as you can see, but again, they are explained. So the action potential. When sodium comes into the cell, there is a series of graded potentials, which means a little bit of depolarization. It goes up, kind of like the last one that you saw, the last slide. If there's a couple more sodium molecules to come through those channels, it goes up a little higher. Only if it reaches the threshold will it actually spike up. Why is this? What is so special about the threshold? Well, the threshold is special because it has a certain level of voltage. It is at around -60 millivolts. And most of those protein channels, remember those are kind of like gates that can be open and closed. Most of those protein channels on the cell, on the neuron, are what we call voltage gated, which means that when the cell reaches a certain voltage, those gates open automatically, kind of like a remote control that opens them as soon as they reach a certain voltage. My air conditioning sometimes goes on when the temperature reaches a certain temperature. It's the same idea. So when we get to the threshold at -60 millivolts, what that does is it basically opens up every single sodium channel on the neuron. And if the sodium channels are open, does sodium want to come into the cell at all? Well, let's think about that. It's got diffusion, pressuring it to go inside, and it's got electrostatic pressure. So it really, really wants to get inside. So once the sodium channels open at the threshold, sodium rushes in at 500 times its normal rate, causing that huge spike in voltage that goes above zero all the way up to like positive 30 or so. When we reach the peak of the action potential, at one point, sodium has basically done all it can do, and the sodium channels then inactivate automatically. During that rising phase of the action potential, once the voltage goes past 0 into positive, guess what happens? So we talked about potassium being kind of motivated to get out because of diffusion, but it was really kind of comfortable also because of electrostatic pressure. So potassium kind of wants to get out, stretch its arms out, right? Stretch its legs, but it's not really all about escaping the cell. Not until it has more than one of these forces at work. Well, when the cell becomes temporarily positive because so much sodium is rushed in, guess what happens above 0? That's right. Now potassium has both of those forces. Now it's always had diffusion, and now it's got electrostatic pressure because temporarily it's positive in a positive cell. The cell is now more positive inside than outside. And so that's when potassium says, okay, now I want out. So the difference between sodium channels and potassium channels is that sodium channels open really quickly. They open and close, activate, deactivate in the snap of a finger. Potassium channels, they're kind of old, like an old garage door. So they open very kind of slowly. So when the voltage gets past 0 during this rising phase of the action potential, potassium channels slowly start to open. But again, potassium can't get in or out until they're fully open. So at the peak of the action potential, around positive 30, sodium channels inactivate, so no more sodium can come in. And potassium channels are now fully open, so there's only one way for this voltage to go, and that's back down, because there's only one thing happening here. Sodium is inactive. Potassium is now leaving the cell and bringing that voltage level back down towards resting potential again. So if I am allowed to go back before it probably will kick me out of this if I do that. The voltage gated potassium channels open above 0. And then once we get to the peak at positive 30, they're fully open and potassium exits. Oops, I'm gonna just do this manually because my computer's a punk. And if I go back to this chart here, because I want to show you guys what it actually looks like. So here is at the threshold. This is sodium channels going above 0 and rushing in at 500 times the normal amount, right? Once it reaches threshold, all of those voltage-gated sodium channels open, and sodium whoosh rushes right in. During the rising phase, that's what's happening. 500 times its normal amount, sodium is just rushing in. Once you get above 0, that's where those potassium channels are opening, but relatively slowly. So they don't fully open until we get to the peak here. At the peak, two things are happening. One, sodium channels are inactive, they shut off, and potassium channels are fully open. So the only thing that can happen is potassium going out of the cell, causing the voltage to go back down. Once we get back down around the threshold level, then those sodium channels begin to reset and can be active again, but voltage level is still falling down. At this point here at resting potential, the potassium channels now are starting to close, but again, they close really slowly. So here at the threshold, sodium channels are active again, but the voltage is going the wrong way, so it will take a lot more to get another action potential. Here, at the very bottom, what we see is that potassium channels have now fully closed down here. We call this the undershoot or hyperpolarization phase. And then eventually, the sodium-potassium pump brings everything back to normal, and that's our action potential. Now, that this whole thing takes place about 1 to 2 milliseconds, 1 to 2,000th of a second. I shouldn't have done that. I should have gone to the next slide. So allow me to do that. And I apologize again for technical difficulties. So here we see that movement of the action potential down the axon is not a flow of ions, but it's a chain of events. When the action potential reaches the terminals, it passes the message on to the next cell in line. So once that electrical activity gets down there, it causes the chemicals to bind to the membrane. It opens up the membrane, kind of like opening the gates or a garage door, and it allows the signal, it allows the chemical to flow through that synapse, through that gap, and be picked up by the dendrites of the next cell. This local potential is called a graded potential. And the graded potential, if it doesn't reach threshold, is going to get pumped back out, and you go back to resting potential. But the action potential, if you reach the threshold and it opens all the voltage-gated channels, you get an all or none phenomenon. As long as it reaches the threshold, you will always get an action potential. If it doesn't reach the threshold, it's going to stay local or graded, and it's going to flatten back out to resting potential. So the action potential is always going to look like that voltage-wise. It's always going to occur at full strength. It doesn't vary with stimulus intensity, which means you can't get a bigger or smaller action potential. It will always be the same. And the fact that the myelin sheath is on these axons also means that it's non-decremental. It will never get smaller in size because it's always recharging itself and sort of jumping from bare spot to bare spot. So no matter how big or small the axon is, your signal is going to be just as strong at the end to release that chemical as it was when it started. So the message travels over long distances at the same amplitude. And the last thing I'll go over in this half of the lecture is that how do we know when a signal is stronger or weaker? It's not a bigger or smaller action potential, but it is more action potentials or less. So if a light looks brighter to you, like a really bright light, you have more action potentials in the same amount of time. If a light is really dim or dull, you have less action potentials. If a sound is really loud, more action potentials. If the sound is quiet, less action potentials. That's how our brain can compute these things. And of course, it gets the signal from the chemicals we call neurotransmitters. So let's stop here for now. And the next half of this lecture, we're going to talk about what happens chemically when the signal gets to the end. And we'll talk a little bit more about things like threshold, refractory periods and things like that. So stay tuned. I'm going to give you guys a mental break. And as always, I'll talk to you soon.